All right, hello everybody. So I'm going to go through what I'm expecting for this mitosis/cell cycle verbal. So you will have this checklist in front of you. I know it's hard to see, but it is the same one in your class packet. And you'll have your chalk and your foam manipulatives. So you're going to have this checklist. I'm also going to ask you guiding questions in case you finish one. I'm just going to go through and check them off as you do them. So I'll be like, okay, show me what a resting cell looks like with 2 pairs of homologous chromosomes. So you're going to draw a nucleus out of chalk, and you're going to show me your 2 red and your 2 blue chromosomes. The red and the blue are homologous to each other, meaning they are very similar to each other. They just have different versions of the same genes. So now it says state how many total homologous chromosomes are in each human cell. So this one we only have 4. In human cells we have 46. So I'm expecting you to say 46. And then you're going to show the chromosomes within the chalk nucleus. We already done that. Boom, good. So next phase, interphase. This happens before mitosis. And that is when the chromosomes are replicated or doubled. So I'm just going to go ahead, take my other foam pieces, and attach them to the same colors. This represents the DNA because remember these chromosomes are just made out of DNA, just really tightly wrapped. And they're going to double like this, where they're attaching at the centromere, which is this inner part. All right, so our DNA has doubled, and then it says label the sister chromatids and homologous chromosomes. I'll just have you tell me which one is which. So your homologous chromosomes are just like what we talked about at resting. It is the red and the blue to each other and the red and blue to each other. Sister chromatids are completely identical. So these are going to be the ones that happen after replication. So the blue and the blue, the red and the red, blue, blue, red, red. These next to each other are sister chromatids. Now we still have 4 chromosomes. They each now have a sister chromatid. So then we move on to prophase. We state what happens to the DNA. So you can't model this with the foam manipulatives, but we know that DNA is normally like long and stringy and uncoiled. So in this step, they actually coil up together and condense into these chromosomes. And then model what happens to the nucleus. You're just going to erase your chalk nucleus because in prophase, it disappears. And just remember, it disappears because if it were still there, only the nucleus would divide, not the entire cell. So next we have metaphase. Metaphase is one of the most important steps because they line up in the middle of the cell. Doesn't matter if you do red, blue, or the order of the colors. It just has to look like this. All right. Let me push it that way a little bit. It's a little off camera, but you get the gist. And we state why this step is important. Well, it's important because if they don't line up in the middle, we have no way to split them equally to opposite ends of the cell. So they line up in the middle to make it easier to pull apart. This brings us to our next step, anaphase, where all these spindle fibers, which are these yellow things, attach to the centromere of the chromosome. So we just attach these to all of them. Touch these, touch these. Hallelujah. All right, then after that, we have anaphase. So this is where the chromosomes pull away from each other. So anaphase starts with an A, they move away. So you pull them by the spindle fibers because that is what is controlling this. Spindle fibers are essentially just like strings that pull them apart. Okay, so now we split, and these are going to go into your 2 different cells. This step is the most important because this really ensures that all DNA is equal in the 2 cells it produces. So now that moves us to telophase. That's where the spindle fibers disappear. So you'll get rid of these. You'll state, because again, we can't model this, that the DNA uncoils or unwraps. And this DNA, it's no longer going to be in a line like in metaphase. It's going to condense like this again. Telophase is kind of the opposite. Oh, here. Telophase is kind of the opposite of prophase in that the DNA, instead of condensing, decondenses, and the nuclei is formed. So you're going to draw 2 new nuclei around each set of DNA. So now the very last step is cytokinesis. This is where the cell actually splits into 2. So I'll have you model this just by drawing another circle around the nucleus to represent the cell on both sides. Okay. And then you'll have to state the end products of mitosis. Well, look, there are 2 identical cells that are the same as each other and that are the same as the starting cell. So these are called 2 identical daughter cells. And you'll state the purpose of mitosis. The purpose of mitosis is specifically to repair our cells. So if you get a cut, this is how we repair ourselves. And it's also to grow. This is why you don't stay babies forever. All right, good luck.